Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Can you guys hear me? Doesn't seem to work. Testing. Can you guys hear me?
Yes, Doctor.
Okay. Alright. Something is wrong with my mic. One moment. Right. I think this is better. Okay. So some of you guys are still coming, but that's fine. We will. What happened to the slides? Okay. Right, back on track. Sound check. Okay. Okay, so Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and a very good day to everyone. So today will be our last lecture. Okay, even though it's only week 13, but it will be our last lecture. Next week, you will have your alternative summative assessment for this course, SID3020, starting from 12 p.m. like now. For 24 hours. Okay, so you need to submit within 24 hours. I'll open up a link for your assignment submission on Spectrum. So in case if there is any issue with Spectrum, then do let me know so that I can open a different link, or I will suggest a different ways for you guys to submit the assignment. Okay, so either WhatsApp or email and whatnot. So I will decide on that in case if there's an issue. But say if there is no issue, then Spectrum will be our main platform for the assignment submission, for the assessment submission. Okay, it's not assignment, it's more an assessment. But before we go into a bit more detail, so we'll just focus on our last lecture. It won't take long. I'll plan it for like half an hour. Then we will have a Q&A if you do have any questions that you want to ask. Then today will be the last day where you can ask me. Otherwise, you need to ask via WhatsApp or email, which might or might not take some time for me to answer. Okay, so lecture time is the best time for you if you have any concerns and you want to ask. Okay, so today's topic is about cellular technology. If you look at previous, or if you recall previous lectures, sometimes I put in vivo. Okay, in vivo technology. So that terminology is not the most accurate ones, because in vivo can also means in a living animals, for example, or living humans, animals, and whatnot. So I change it to make it more accurate, which is cellular. Because what we are trying to do is to compare an enzymatic reaction whereby you take out the enzyme from cells, compared to if you are using cell itself to produce whatever you want to produce. Okay, so that is a more accurate terminology. Just bear that in mind. For today's attendance, if you have not yet submit your attendance, you can just QR this code quickly. And today's code is cell tech. Okay, so we've looked at a lot of enzymatic technology previously. For the past few lectures, we've looked at how enzymatic reaction works, how can you produce it, how can you isolate it. So today we're going to look at a little bit more about whole cell. Okay, so what are the difference between enzymatic and a whole cell biotechnological approach? So if you consider this reaction on the right hand side, whereby initially you have one small molecules, and finally you will produce a fat, okay, which is a very huge molecule. If you consider you are doing an enzymatic reaction, just for the sake of enzymatic, so we are not considering chemical yet for this image. If you're just focusing on just using an enzymatic reaction to produce fat, you can see that they are actually 5 different steps or enzymes, even though some of them are similar, like those ones. Okay, 1, 2, or I missed 3. Okay, there are 5 different steps where you need to use enzyme, and then you do a stepwise, kind of like a stepwise reaction, but using enzymes. So it will be too much work. Okay, it will be too much work to actually do each reaction using enzymatic reaction, because each time when you do a reaction, you need to purify it. Okay, you do that one, then you need to purify. Why you need to purify? Because sometimes, even though it's not always the case whereby you need to purify um, on every reaction, but sometimes what might happen is that two different enzymes, even though they belong to this to a different category, they might influence the reaction altogether. So it's always best, of course, if you optimize the whole reaction and whatnot, then everything over here can be in one pot. But in case if you're doing it one by one, then you need to purify on each step. So to do that, it will take a lot of time, a lot of energy. Therefore, the best way to actually do it is by using a whole cell. Okay, so if you look at this question on the left hand side, isn't it too much work to purify the product after each steps because before proceeding to the next step? Then the answer is yes. But of course, as a chemist, you might say no, because towards the end of the day, if you're thinking about just the chemistry perspective, you can quickly produce a fatty acid without using an enzyme or whole cell. Okay, but because this course is to look at the biological perspective of synthesis, then this example is actually not the best example, but nonetheless, it is still an example whereby you can compare. If you're doing a chemical synthesis, then it will be very easy, very straightforward. But if you use enzymatic reaction, you need to have different enzymes. But if you use a whole cell, then you can just do it in one pot reaction. Okay, so using cell to do the work can fasten the steps, of course. So instead of doing all these 5 steps, including all the purification, you can just quickly convert from the reactants. You put in everything, all the reagents that you want, then put it in a cell buffer, cell media and whatnot. Then you can quickly produce a fatty acids. Okay, and of course, alternatively, if you just want a fatty acid, like is shown in this reaction over here, you can simply grow cells, kill it, and then just extract out the fatty acid. Okay, so there's a lot of ways to actually do it, but this is just an example whereby a different approach can be used to actually achieve the same outcome, which is to produce a fatty acid. Okay, but again, it's not as easy as it sounds. Why is it not as easy as it sounds? Because doing a whole system cell-based biotechnological approach, there's a lot of things that you need to know. There's a lot of things that you need to consider before actually having an outcome as shown in this image. So in order for you to do that, what do you need to know? So there's a lot of things. The field is very huge, and I think it's actually not really I'm not going to touch everything, of course, because you are all chemists, and this is just an elective course. So it's best for me to just give you an overview of what you need to know, a little bit of idea how you can use an enzyme or a cell-based assay to get what you want. But towards the end of the day, if I were to tell you like a very detail, you might not use it at all. So today's lecture aims for just very basic about cells technology, and then we can compare with a biochemical or enzymatic approach. Okay, so those are just the aims. So as long as you can grab this concept, then you will do fine. Okay, because to the end of the day, in the future, if you are working with a biotechnological approach reaction, then you definitely need to read, because of course, if you're just focusing on the cell-based technology, it can actually be one course on its own. Okay, so because I just want to cramp it into one lecture, then it's very basic here and there. Okay, so this is an example, a picture of a cell, okay, a normal cell. If you have not taken any biology, so that is a cell. That in the middle there is the nucleus, which is kind of like the brain of a cell, even though it's not really a brain, but you know, it's kind of like a brain of a cell. In the middle, the yellow ones in the middle is a nucleolus. So this, you don't need to know in very detail what are each organelle's function as, but this is just an overview so that you guys know a little bit about cells if you have not taken any biology course. Okay, so that's the nucleus, that's the brain. So this is where we have our DNAs, and this is where our RNA is being produced. So the RNA vaccines, COVID and whatnot. So this is where cells actually produce RNAs. Okay. Surrounding the nucleus, the one in the yellow is the Golgi. So we call it Golgi Apparatus. So this is where post-translational modification from RNA to protein is being made. Also, so these two places, Golgi and endoplasmic reticulum, that's what we call it, is where protein are being synthesized. Protein are being synthesized. There are a lot of post-translational modification that is being done on it, so that it can be a functional protein or enzymes. Now, why do I need to talk about that? Because towards the end of the day, to know how you're going to proceed with a cell technology, you need to know where is the enzyme of interest are located. Okay, so you need to know where they're located. Are they located as an extracellular component? Extracellular means either it's embedded on the cell membrane over here. Okay, so that one is the cell membrane, or the enzyme is actually being excreted out, meaning that once produced by the ER, the enzyme being transported out of the cells. Okay, so you need to know whether it's embedded in the cell membrane or exported out from the cell membrane. Additionally, you need to know whether the enzyme are actually intracellular, meaning that the enzyme are located inside of the cell and actually not being transported out of the cell. So by knowing either these two, you can select an approach. Okay, so the approaches mentioned here is either to use an enzymatic approach or a cellular approach. Okay. All right. So to know which approach that you need to best utilize, then you need to know where is the enzyme allocated. Okay. Again, if the enzyme allocated as an extracellular component, then either approach is fine, meaning that you can use an enzymatic approach where you extract out all the enzyme very, very easily, very, very quickly, and then use it for your reaction. Or you can also use cells to perform the reaction that you want. But if the enzyme are actually intracellular enzyme, meaning that it's inside of the cell, then the only approach that you can use, of course, you can still use enzymatic approach, but cellular approach might be better in the sense that it will be easier to just grow the cell, put in whatever you want, and then the cell will produce the product that you want. So an example. So previously I've shown you the pathway for fat synthesis, right? So if you were to extract out the enzyme, because fats are being produced inside the cell, and then it's transported out to the cell membrane. Okay, so it's produced inside here, and then it's being transported out into the cell membrane. So if you were to produce a lipid as shown in the graphical in the reaction scheme just now, then of course if you want to produce a lot of fats in a very short time, then using cellular approach is better because the enzymes are functioning inside the cell, and then towards the end of the day the cell will produce the fat on the cell membrane, and then you can just quickly kill the cells, extract out the lipids. Okay, I hope you understand what I'm saying here. So to extract out individual enzymes here, okay, you have 4 enzymes based on the previous reaction. You have 4 enzymes. If you were to extract individually 4 enzymes and then conduct an enzymatic reaction for the lipid synthesis, then it will take a lot of time because each enzymes you need to isolate, purify, isolate and purify. Okay, so you need to isolate and purify individual enzyme before you can actually run the reaction as shown in the schematic on the previous slide. But if you use a whole cell, what you need to do is you need to just have one cell, just a single cell is enough, with all the medias that are needed so that the cell can actually produce more cells, and towards the end of the day, you can kill the cells, extract out all the fats. Okay, so those are 2 different approaches. So then that is what this slide is all about. So this slide is pretty much the most critical slide to categorize what are the options that you have and what is the best option that you can do. Okay, so I've already talked about it. So should you want to do a reaction with path, should you choose? So enzymatic path or the cellular path? Again, the answer is the location of the enzyme. Oh. Okay, so just a summary, comparison between extracellular or intracellular enzyme. So again, based on the location of the enzyme. So if it's an extracellular enzyme, then you can choose either options, either path, okay? But if it's intracellular, then cellular biotechnological approach are the preferred way compared to an enzymatic reaction. And these are some of the reasons. So extracellular, you can use either because the enzyme isolation is very simple, it's very quick because it's outside of the enzyme. It'll be very quick to actually isolate them. It's a very typical enzyme is low selectivity, meaning that there's no restriction requirement to use the extracellular. So either you take it out, it will still function. Even if you put it, use a whole cell, it will still function as what you want. But in comparison, for an intracellular enzyme, they might have a specific requirement. For example, it requires a specific pH to actually work, or it might require a specific coenzyme or catalyst that is available inside an enzyme to actually work. So there's more work that you need to do if you were to extract the enzyme out of the cell and then use it in the lab. Okay, that's what I mean by potentially high selectivity. You might need a specific environment to actually work in an optimal condition. Okay, and additionally, the last point is that extracellular enzymes are normally very cheap. Okay, it's very cheap to produce, it's very cheap to purchase. Okay, compared to intracellular, it's more expensive, and normally intracellular enzyme, the quantity is very limited in contrast to an extracellular enzyme. Okay, so recall this slide that was shown, if I'm not mistaken, in our first lecture. Okay, so biotechnological approach, what's important is the process. Okay, it's the process or the flow of what you want. So in this figure, it's shown 3 different approaches that you can do. You can either use a genetic technology whereby you manipulate or you edit the DNA in the nucleus so that it can produce whatever you want. Okay, in this case, normally what you're talking about is to produce an enzyme that you want. So you can modify a cell so that it produces the enzyme that you want. Then you can use it. Or the second option is a fermentation, whereby the cell has already contains the enzyme of interest, the enzyme that you want. So you can simply just use the cell, you take a substrate, fermentate the substrate and the cells, and then the cells will process and produce the products or the materials of interest. Okay, and as a comparison, enzymatic technology, we've touched it a lot in the previous lectures. So this is not of our interest. Okay, so we're going to focus more, a little bit more on the cellular technology itself. Okay, now, how do you proceed? So for cellular technology, how do you proceed? It depends on which option you want to opt for, whether it's genetic manipulation or fermentation. So you have two options. For fermentation, normally you already know the process of interest and the cell that you want, meaning that the cells here can already produce the enzyme, okay, the enzyme of interest. And you know that the cell can produce, say, for example, if you want to produce A to B using cells, you want to know that the cells can actually produce B. Okay, so if you want to proceed with fermentation, then this is the part that you need to know. And then once you know that the cell can produce the product of interest, then what you do is you simply have the cells in a media. So media here means the growth media for the cells to actually survive. Okay, so basically something that you need in the lab is such as glucose as the energy. You need to have a certain buffer. So depending on which cells you want to use, so some cells use a different buffer. Some can actually grow in PBS, phosphate buffer cell line. Some requires a more slightly different pH, a different either phosphate pH or a different buffer altogether. And of course, normally in the media, it contains an antibiotics or an antifungal so that you don't grow bacteria inside your cell. Okay, so together with the cell, you don't want to bacteria or fungus to grow, because to the end of the day, these bacteria and fungus can kill the cells and you won't be able to get your product. Okay, so this is the first thing you need to have. You need to have your cells, you need to have your media, and then you put in your reagents, whatever you want. Okay, so the reagents, of course, depending on what are the process involved. Okay, if you're just talking about A converting to B, then of course you need to have A as part of the ingredient. What you normally do in this process is first you grow the cells and the media until it reaches a certain numbers. For example, initially, if you only have, say, 100 cells, okay, and of course, if you were to do a reaction using 100 cells, it's very slow, okay? So normally what you do is you grow the cells to thousands and thousands of cells, and then once it reaches a certain number, then you put in a lot of your ingredients. In this case, for example, you put in a lot of A's, and then very quickly you can produce a lot of B's, okay? And of course, what I mean by quickly depends on the reaction itself. Some reactions requires a long incubation time or fermentation time, while some requires like a very short reaction. This, of course, depends on the location of the enzyme. If the enzymes are being excreted out of the cells, normally it's very, very quick. If the enzyme are located on the cell membrane, it's moderate, not too fast, not too quick. But if the enzyme are actually inside the cell, then it will take a longer time. It will take a longer incubation time, because you need to make sure that A goes into the cell, converted into B, and then hoping that the cell will excrete out B, or otherwise you need to kill the cells to actually extract out B. Okay, and that is the isolation process. Of course, I'm not going to go into detail because there are so many things that you can do, so many different types of isolation that you can actually do. So I'll just stop at isolation. And of course, when I stop it here, meaning that it won't be in your exam. So you just need to know that you need to isolate. That's it. All right. I wonder why I still have that. Okay, so in case of genetic manipulation, so it will be slightly different. because first you need to have the gene or protein of interest um, that will result in the process of interest and the cell that can host the gene. Okay, so there are three different components because they are actually linked to one another. So first off, if you're talking about a cell that does not contain the protein of interest, okay, so that is what genetic manipulation is all about. Genetic manipulation is originally the cells does not have what you want, so you need to manipulate. You need to edit the genetic material first. So that's why a gene of interest is mentioned here. So you need to have a gene of interest that of course the gene will produce a protein that will result in a process of interest. Okay, and then what you do is you put in, keeps on changing the, okay, what you do is you put in the gene into your cells nucleus. So of course you need to transfer the gene inside the nucleus so that the cells can now produce the enzyme of interest. Okay, so you have your gene, you put it in, now it will produce cell produce, so let's call it gene X. Okay, produce protein X. Okay, now once the enzyme, once the cell has produced protein X, then you can proceed with the process of, say, converting this time around probably C to D. Okay, initially without genetic manipulation, you cannot convert C to D. So if you put the cells with C, nothing will happen. But once you have the gene of interest inserted into the cells, and once the cells has expressed the gene to produce proteins or enzymes, then now if you put in C, C can be produced into, can be converted or processed into D. Okay, so the process of insertion of a gene into a cell will produce the cell of interest. And again, the steps is more or less the same. What you need to do first is you have your gene, you put into cells, okay, and then the cells can be used, can be grow even further. So of course, when you put gene into cells, if you start off with 100 of cells, towards the end of the day, maybe just 50 of the cells contain the gene of interest. It's similar as in you are doing a chemical reaction. So you have 1 mole of A, you want to react with 1 mole of B, you want to produce 1 mole of C, but sometimes that does not happen. What you will get is, say, 80% or 70% of yield. Okay, so similarly in biology, each process that you do, you normally don't get it to convert to 100%. So to include a gene X into a cell, if you start off with 100 cell, towards the end you might have like 50 cells, and then from these 50 cells, you put it into your media, you grow it so that you can get more cells containing the specific enzyme or a specific enzyme or protein X. Okay, and then from there you can put your C reagents. You repeat the same process where you incubate. And then the cells will produce more products, and then finally you isolate the products. Okay, so those are basically the general overview of cellular technology. So you either do based on fermentation approach, or you do based on genetic manipulation approach, but either or, both are a cellular technology. Okay, but again, by no means what I've shown you here is the only way. So in case in the future you are working in a company that wanted to utilize an enzyme or cells for a specific purpose, then of course you need to do a lot of readings. You need to open up the literature so that you can find the best way to actually achieve your goal. Okay, over here I did not talk about bioreactor and whatnot. There's so many things that you can actually talk about cellular technology, but I'll just skip it because I think it's you know, it's not part of the what you need to know. So the course asks us to teach you about biotechnology. So these are kind of like the biotechnology. It's on the surface, okay? By no means these are very specific examples. Okay, and finally, enzymes of cells. So that one is an isolated enzyme. That one is a whole cells. So from over here means the source. The source can be any source, dissolved in water, suspended or immobilized enzyme, or form. Sorry, not source, form. So the isolated enzymes can be in either of these forms, and whole enzyme can also be in any of these forms, and there are pros and cons for each forms. So this is also critical for you in the future, for you to know, what would be the best option that you want to achieve. Okay, you can just read on your own time. It's very straightforward. I don't think you need any extra explanation on this. Okay, so that one is just an example of top right image is an example of enzymatic reaction. If you were to do it using an isolated enzyme, normally this is how you do it. You will do a reaction in a very, very small scale. Of course, if you are working in the laboratory, if you are working in a company, so you might have something like that, which is a bioreactor. Okay, so this one in the middle here. But bioreactor also can be used, well, depending on the types, of course. The term bioreactor is very general. There are bioreactors that can use for something else, multiple things. But a bioreactor is a bioreactor nonetheless. So you can use a bioreactor to produce, to have an enzymatic reaction, as well as to grow cells and to have cells to produce whatever product that you want. Okay, and I think that's all. So for this course, the learning outcome for this course, especially for my section, is number 1, 2, and 3, and I believe that I have covered all 3. So I'm hoping that for now you can actually classify natural products according to the structure and metabolic pathway. So not so much on the metabolic pathway, but more on the natural products according to the structure. So in that sense, meaning that you now know what are the structures of proteins, okay? And then from the protein, you know the structure of a primary structure, secondary structure, tertiary structure, quaternary structure, the differences between those two, a general structure about how inhibition works, about cofactors and coenzymes, all those things, okay? And then number 2, you can apply appropriate extraction and separation method for natural products. Of course, in this case, we are focusing solely on the enzymes. Okay, so you know how you can now produce an enzyme, even though it's very brief, of course. By no means this course is to go in very deep. And you know how to extract, the process of extraction, what are the general steps, the basic steps that you can use to get the enzyme, to isolate the enzyme. Of course, if the methods that I've told you previously did not work, you need to go into the literature, find a more specific steps for the enzyme of interest. Okay, so the ones that I've shown you previously is just for a general ones. Number 3, that one is more on Prof. Khalija's section, whereby using spectroscopic data to actually identify all these natural products. And number 4, to illustrate the use of biochemical and biotechnological methods in chemistry. So this one, again, the terminologies used here is biochemical and biotechnological. And my previous lectures are talking about enzymatic technology, cellular technology, fermentation, this and that, okay? So some of you might have this question: what are the difference between biochemical and biotechnological approaches? Okay, so what are these two terminology means? So I have a few slides, just 1 or 2, to actually kind of like divulge the terminology so that you won't get confused in the future. Okay, so it's basically just a terminology. A biochemical are basically using an enzyme for a chemical process. Okay, so even though bio there can mean a lot of things, cells and whatnot, but for this course, biochemical approaches mostly referring to using enzymes for a specific purpose. And therefore, a biotechnological approach are the other ones, which is using cells for a chemical process. Okay, so those are just the technology. So you need to understand both concept. Then you will be able to achieve the linear outcome 4 of this course. Okay, what do I mean by that? For example, if in the final exam, okay, if in the final exam has a question something like this, using biochemical process, illustrate how you can utilize polydioxyribonuclease synthase or ATP enzyme in a chemical reaction to produce compound A. Okay, so compound A is that one on the top, on the right hand side. Okay, so your answer should include something like this. So what is defined as the chemical reaction? Okay, so over here, what are the chemical reaction? So you know that's the enzyme. Okay, so you know that's the enzyme. Then you can, using the biotechnological tools, the website that I mentioned in the previous lecture, you can find out what are the the chemical reaction involving the enzyme. Okay, so what is the function? Of course, to know the reaction, you need to know the function, whether it's hydrolysis enzyme, whether it's transferase enzyme. So the seven categories that we've touched in lecture number 3 or 4, I can't remember. Okay, so you need to know the function. And then you need to know what are the reaction look like. Okay, so that reaction look like is, of course, the ones that I've shown over here. Okay, and then where can you source the enzyme? So those are the things that you need to know. In case if a question like this comes out. Okay, I'm not saying that it will come out, but in case if it comes out like this, so these are the things that you need to know. And from there, you need to illustrate all of this. Again, for a question like this, I will probably put between 5 to 8 marks. Okay, because for each category that you put here, you of course need to have a little bit of explanation, and the explanation you don't need to like make it very long. So the exam reflects on the quiz that we done, two of the quizzes that we've done previously. If you notice that all the subjective questions, I always put a word limit or a sentence limit. So that is actually to practice, to make sure that you guys know that you don't need to answer like very, very long, a whole page just to get 5 marks. Okay, as long as you can cover the terminologies that is being used, then you'll get your marks. As simple as that. Okay, so something like this will have about 5 to 8 marks, and if you just answer like that, so it will just be like maximum of 10 sentences. So you don't need to write like a whole page to get these 8 marks. So please bear that in mind, and always look at the marks for each question to actually know how long you need to write it or how long you need to give your answers. Okay, so this is just an example, and of course, this is a biochemical processes. It will be the same for biotechnological processes. You need to understand all of these. You need to understand how you achieve that, so that you can explain it in a better way. Okay, so relate everything back that you have learned about enzymatic technology and then put it in. Then, of course, if you do that, you will not have any issues in getting any marks. Okay, and that's it for today. So these are some of the main references that I've used for the past 13 weeks, -2 for the quiz, so 11 weeks. So you need to accustom yourself with Protein Data Bank, like how do you use it, and additional readings. Of course, you need to go back and look at all the previous lecture notes. Make sure you understand, make sure you follow through the processes and whatnot. You can also look at my website. So I do have different lectures about this and that. So all my lectures I actually put it in my website as well. And of course, for the latecomers, make sure you sign in for your attendance. Okay, and today's code is CellTech. I'm hoping that everyone attended all 3 synchronous lecture, because otherwise we might have some issues with your final exam. Okay, as you know, previously you need to attend like at least 80% of your attendance before you can actually sit for your exam. But so far I did not block anyone from attending your exam. But in case if there's any issues in the future, then if someone opens up the attendance and then see that you guys did not put in or sign in for your attendance, then there might be some issues. So that's why even Prof. Halija has always reminded you in your email, please sign in to your attendance and whatnot. Okay, I think that's it for today. If you do have any questions, we have 9 more minutes. Otherwise, thank you, and I will brief you on your exam. Or probably there's no briefing. Oh, it's very straightforward. It's kind of like your previous exam. I will just give you the exam questions, and then you need to complete it within 24 hours. Okay, so that's it. Thank you. You may leave now if you don't have any questions. But if you do have any questions, by all means, unmute and ask them, or you can just put it in the chat. I will stay here until 12:50. Thanks, guys.
Thanks, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.